And now for something completely different. Why? Also warum? Warum zieht jemand aus einer der anerkannt schönsten Städte oder lebenswertesten Städte der Welt, why is somebody moving from Vienna zur Perle des Bergisch-Rheinischen nach Bergisch Gladbach, ist schon klar, aber warum tut man sowas? Ja. Aus Wien hierher. Der Liebe wegen ist es meistens, ne? So, bei ihm auch, because he was falling in love with science, mit der Wissenschaft. Und zwar auch dann noch mit irgendwie diesem bildgebende Wissenschaft und Bildgeben in der Medizintechnologie. Ja? Und damit hat er sich befasst, macht sein PhD oder hat sein PhD gemacht und wir hören jetzt ein bisschen was über Maxima, ne? Und Bildgebung bei uns und der Benjamin Baucher aus Wien.
Okay, thank you very much for the introduction. And yeah, well, if science call, one has to answer. There's no other way around it, right? That's just the way it is. But before I start talking about science, I want to take you to a much nicer place, to be perfectly honest. I want to go on a trip. I want to go to the beach. So I want you all to imagine you're at a beautiful place. The sun is shining, the sky is blue, the sand beneath you is soft and sweet. It's beautiful. And so you look out at the horizon and you see many islands that are across from you. And since it's so nice today, you decide, okay, why don't I go explore these islands? Why don't I have a look around and see what's happening? So here, I've drawn a map. I have to say, I'm not an artist. This is not close to what I've just shown you, but let's just imagine that this is the place you're at. These are all very nice islands. The sun is still shining, the sky is still blue. This is beautiful. So you will first see all of these islands. You will first see a sketch of them. You will notice where they are in location. You will see what they are shaped like, which one is behind which another one. Then you can look at them, you can study them a little bit more closely. You can see, okay, this island that we see over here, for example, this one has volcanic soil. It might be dangerous to go there. Maybe I don't go there unless I really have to. This one right here is nice. This is supposed to be a very nice green color. For some reason, it is blue now, but I'm not going to complain about this. So this is where, for example, agriculture could be. So this would be a nice place, for example, to go for a hike. And down here, this one right here, this is a nice gray, nice brown place where there's a lot of mountains. And as we just heard, I'm from Austria, so mountains for me are perfect. So this is the place you want to be at. And so just by looking at these islands, you can draw conclusions on what they are. You can see what they are doing, and you can also maybe even draw conclusions on what they're doing. So this is, for example, you can then say, okay, this island has some volcanic soil, it's dangerous, I might not go there. This island right here, agriculture. This is a very nice place. It's producing stuff for the islands around it. Everything is interconnected. This here is a mountain place. There's probably not that many people there, but there's still, for example, tourism that might be interesting to do over there. And this is exactly the same as what happens in microscopy. We look at the tissue samples from the body. We just study the cells that are in there, and we make conclusions based on what is in there. There's, of course, different modalities than just microscopy, but since I like images, I want to focus on this one. And here I have a very, very nice new modality to work with, a very nice instrument, which is the Maxima. Now, we're all here from Miltenyi, so I probably don't really have to talk too much about it, but still, I just want to mention that this is what's called a highly multiplexed immunofluorescent microscope. And this is, of course, an amazing term that captures everything. I don't need to say any more, but I'm still going to. And the idea behind it is we can take out, for example, a tissue biopsy from a tumor. We can put it into this machine, and then we can look at this tissue sample with a bunch of different fluorescently labeled proteins, so we can see exactly which cell expresses which protein. This is, of course, very important if you're studying immunology, especially in the tumor context, because you really want to see how everything is organized. However, there is one big difference between what I'm showing you here and those islands right there, which is that when I first led you to these islands, I told you you can see them, you can just look at them, and you know exactly what each island entails. You can just look at this island and say this one has volcanic soil. Here, you just see colors. And you have to know first that, for example, this roundish shape right here is one particular cell. So you don't even start off with an idea like this, where you have this beautifully well-defined island, where you know exactly where one ends and the next one begins, but you just start with a question mark. And so this is where my work begins, because we want to somehow automate finding all of these cells that are in this very beautiful, colorful picture that I've just shown you. And this is very, very challenging. And it leads us to a very dark place in data science, a very difficult place that requires a lot of computation, and that is called computer vision. Here I have the official definition. Computer vision is a field of artificial intelligence that uses machine learning and neural networks to teach computers and systems to derive meaningful information from digital images. This is to say, this is AI. Now, AI, of course, I could go on an enormous rant now about how the term for AI is being misused at the moment, because everything nowadays is AI. My fridge sends me a message on my phone because it is empty. AI. This is, of course, not really what I'm talking about here. I'm talking about actual AI. And instead of going on a big rant about it, I'm going to show you what AI actually means. AI is actually a very old concept. It was first established in the 1950s. And it basically encompasses everything where human intelligence is being exhibited by machines. This is very general. This actually entails the fridge that sends you a message. It is simply, it is cold, it is empty, so I send a message. Same would be a thermometer that turns off the heating in your home, AI. But this is a very primitive AI. AI has many subsets, and these subsets are what are actually interesting. The most interesting one for me personally is deep learning, which is the second one from here. And here the idea is that we have machine learning models, so mathematical models, mathematical systems that mimic the human brain function, that mimic how our brain actually works. For completion's sake, I'm also going to quickly mention generative AI. This is what happens with ChatGPT. So here you have a system that first learns everything about everything it has seen before, and then generates some completely new things. So generates original content. This is very interesting. But for me, I'm much more in the deep learning side of things, where we actually try to mimic what the human brain is doing and try to get the computer to do cool stuff based on that. This is what's called a narrow AI. It has a simple application. Okay, but first we need to start at the beginning, because every single AI system starts with an image, or every single AI system for computer vision starts with an image. And every single AI system, every single computer system needs numbers. So the first question we need to answer is, how do we turn an image into numbers? Well, the answer is quite simple, because images, by their nature, are always two-dimensional matrices. This basically just means that it's a grid that is filled with numbers. Here I've drawn out a nice smiley with zeros and ones, and this is all an image is at the end of the day. It is just a matrix filled with numbers that we can then look at. Then you might ask, okay, it's a matrix filled with numbers, but what about colors? Well, colors basically are just these numbers, what I'm going to call intensity values from now on, colored in. So if, for example, we have a 1, in one of these matrices, we say, okay, this is now the red matrix, and so this is always going to be red. This is called the RGB framework, or red, green, blue framework of saving images, and it's kind of the state of the art. And of course, if we merge red, green, and blue, we get white. If that sounds somehow familiar, it should, because this is exactly how the human vision system works. We have cones for red, green, and blue, which allow us to see color. Why am I mentioning this? Because, again, we are mimicking nature. This is something that computational scientists like to do a lot because we're very lazy, and so we just like to mimic what is already there. And so we do exactly the same when I'm talking about deep learning and actual computational models. And the idea behind deep learning, as I just told you, is to mimic human behavior. And so we want to basically implant the idea of a brain into a machine. So we need to create a model of how our brain works and then put it into a robot, for example. Or in my case, into a computer vision system. So really quickly, how does a neuron work? Well, basically, it's just a transition system. We have an input, in this case, the dendrite. Information comes in this case, it's electrical signals, electrochemical signals, that then get processed by the cell body, that get propagated through the axon, and then get propagated to more neurons through terminals or synapses. And the actual meaning, the actual interpretation of this comes from the amount of neurons that are interconnected. The human brain has over 100 billion neurons. And so this meaning gets derived as we go further and further along these connections, these synapses. And so now, For us computer scientists, we're doing exactly the same thing. This looks a little bit wild at first, but it's exactly the same idea. We have here an input, these dendrites, where information comes in the form of electrical signals. In our case, it's just numbers, as we've seen them from the images. We then do some nice maths in here that happens in the cell body. We then pass it for an activation function. This is just a mathematical construct that we need to use in order to pass it along the neuron. And then we have an output at the end that propagates it deeper into the network. This is now a single neuron. Now, you might think deep learning. Okay, deep learning means we need many neurons. So we start off here with our image. This is just the pixel values. These are intensity values between 0 and 1, for example. And then we pass it into the network. And now this deep learning, where the deep comes in, is that we need many, many neurons in order to actually make something out of this information. This is what's here. I'm sparing you all of the maths that's behind it. There's a lot of it. But what's happening now is we're having this input right here that gets processed exactly as it does in the human brain, and in order to generate an output. So again, we have the same kind of construct of having data that comes in, information that comes in, it gets processed, gets transformed, and then it goes out. Now, how do we apply this for our system? Because we started off at the beach. Everything was nice. The sun was shining. There was what? There was a sea. We need to teach the system we need to teach the AI how to actually do these segmentations, how to find these islands. And you've probably already heard the term training in this kind of context. And training basically just means we show it the same example, we show the same examples over and over and over and over and over and over again, until finally it learns to extract some sort of meaning out of this using all of these connections, all of these parameters that it has accessible to it in the form of its neurons. Again, I'm skipping a lot of math here, but there's a lot of math. Now, with this, we could already get to work. However, this is still too simple for the kind of approach that I'm working with. So what I am personally doing is I'm currently collecting a bunch of state-of-the-art methods that are used for this, which are basically going even beyond this idea of encoding information in a neural network and then doing even more with it. So here, for example, I have a cell-pose algorithm, I have multiple algorithms. All of these are things I need to test in order to finally get to these segmentations. And these segmentations here, I've drawn them in, and these separate each individual cell in these images that we have seen here before. And what we see here as we go through these intensity values on the left-hand side is we see now for each individual cells what is expressing. This, again, is very, very interesting for any sort of immunologist who then wants to know exactly, because now they know exactly what a cell is doing, what protein it is expressing, and also, most notably, where it is in the sample. And is this last step, where is it in the sample in relationship to all of the other cells that has not really been available before. And using this information, we can then go back to this image I showed you before, and we can then do something called downstream analysis. We can then look exactly, okay, this cell is now expressing this protein, this protein, this protein. This means it's similar to this cell that's right next to it, and so we assign it the same class. These are the different colors in this image right here. And this, again, makes the work of immunologists hopefully a lot easier because it brings them to a result much faster because they don't really need to look at every single image anymore manually, but they just run this algorithm, they run the AI, they get something out, and hopefully it's useful. And my hope, my dream, is that then I can go back to my island, I can just run this algorithm, it is able to discover everything for me, and I can just sit on the beach and relax. Thank you.